And hello to everyone. I am delighted to be here. This is a real privilege and thrill for me to have a chance to speak to you and tell you about my experience with developing a metal product and commercializing it and transferring technology from the defense industry, which when I started doing this was unusual. Now it's becoming much more commonplace, but I'll tell you about my experience and all the steps associated with it. You will notice on my charts there is a number in the lower right-hand corner. If you have any questions as I go about anything I've said, if you make a note of that number and something to remind you of the question, I'd be happy to answer questions at the end. Because I have a lot of charts, I would prefer not to stop from any questions unless they're just absolutely critical to interpret something on my slide. I'd rather handle them at the end. First, a few acknowledgments. Some of these Professor Tiles already mentioned. My heritage is in the defense industry. I worked in it from the time I graduated from Caltech, starting at the RAND Corporation. And later in my career, I ended up at Logicon, which was a quite large defense company here in Southern California. And it was there that I led a group that invented the product I'm going to talk about today called Logicon Carriage Detector, and of course that's why Logicon's name is in the product name. Logicon was then bought by Northrop Grumman, and when I wanted to start my own company, I had to get the approval of the CEO of Northrop, and that happened to be Kent Cressa, who at that time, and some of you may know, he was also a trustee of Caltech for 12 years, and actually chairman of the board, so to speak, here for part of that period. And he was very enthusiastic about what I wanted to do. He was really supportive of transferring the defense technology into non-defense applications. I licensed all the technology. I developed a new version. Quite frankly, I'll share with you all, I've never written a line of code in my life. I always hired really good software people, and I had a wonderful team in Taipei, Taiwan. And that may seem strange that they were that far away, but believe it or not, it worked perfectly. We used to have teleconference calls 2 or 3 times a week in the evening because of the time difference, and they did a terrific job. I've handled all the regulatory affairs for this product, both while I was at Logicon in Northrop and then from my own company. And I'm going to talk a bit about the regulatory affairs because I think they may be important to endeavors that are going on here at Caltech. And eventually I have sold the product to Carestream, which is the current distributor of the product. The original distributor was a French company called Trophy Radiology. They were bought by Eastman Kodak, and then Eastman Kodak actually created Carestream by selling their medical division. So that's kind of the corporate background for this. I'm going to talk about the subjects listed here. I think you can read them faster than I can identify them. I do want to get to the updates at the bottom because that involved an exciting new version of this product, which GA Industries is entirely responsible for, and I'm very proud of that development. Background, this work all started at Logicon at the end of the Cold War when Logicon was very concerned about the fact that defense industry business might decline badly. And they wanted to get into other things. So I had a group of people who worked on the subjects that are listed there. And to be very honest with you, we had working software for every one of those. And Several of them were of great interest. The software for the oil refinery was of such great interest to Chevron that they decided not to buy it from me, but they listened to my engineers long enough that they could reinvent it, which always was disappointment to me. But because they're such a private company, when it comes to things that affect their bottom line, they weren't willing to buy software from the outside. And then the same with the third and fourth thing. When 9/11 occurred, that was a really big business. But Northrop, interestingly enough, chose not to get involved in it, and that's just another story I won't go into. But the dental application at the bottom is the one I'm going to talk about today. And it was a very unique application, which we had absolutely no competition for and which has worked out with great success. Why is it important? It turns out that dentists take radiographs to inspect the interproximal surfaces of your teeth to look for tooth decay. And it's the only way they can diagnose the interproximal surfaces. They can't directly visually inspect them and probe them unless you have missing teeth. Then they can, and they do in that case. But it turns out that visually diagnosing film or digital images isn't very accurate. And I have statistics there that were reported from a study done at UCLA that very carefully looked into the accuracy of doctors visually diagnosing radiographs. And you can read the statistics there yourself. The most impressive one is at the very bottom, where 41% of the time they misdiagnose the depth of caries. And that means they may or may not correctly restore a tooth. And sometimes they restore them, as I say there, when there was no decay at all. So this was the motivation for this product. It's interesting in this day and age where you have to disclose everything. I don't know one dentist who's ever told his patient, I drilled into your tooth and it wasn't necessary. There was no decay there. We'll just fill it. But believe me, they do it, okay? And you pay for it, you're right. Let me briefly describe what the device is, and its indication of use is to assist a dentist in diagnosing the depth of interproximal caries. And I'll show you a picture of exactly what that is in a minute. And to determine whether it's deep enough entering the dentin and the tooth needs to be restored. There are some limitations on the product, and we define these very carefully right up front in consultation with Stuart White. His name was mentioned on the previous chart, who did this study. He was head of the dental radiology division at UCLA. Adult dentition was the primary target of interest, and unrestored surfaces, it was not intended to be a concurrent reader. A concurrent reader is something that works independent of the dentist and gives you a statistic as whether or not decay is there. It became very clear when we discussed the nuances of this problem that it would be much better to always keep the dentist in the loop. And Logicon just aided him by giving him additional information. To have something that really replaced him would have been more challenging engineering-wise and much more challenging to get approved by the FDA. Just a quick picture of what I'm talking about. The interproximal surfaces that you'll hear me talk about are the sides of the crown, which are normally in contact with an adjacent tooth. So food gets jammed between the teeth and it begins a decay pattern. The decay pattern literally looks like an inverted triangle, so this isn't just a cartoon. The decay starts on the surface and it expands as it goes into the tooth, so it forms roughly what looks like a triangle. And then when it hits the dentin, it actually, because the dentin is a softer material, a new triangle forms that expands faster. So that's why suddenly there's another triangle there. The dentist is not particularly worried if you have a small site of decay in the enamel. People have them for years, and if they do good oral hygiene, they may never need to be restored. The dentist only worries when it gets close to or touches or starts to enter the dentin, that then you're in trouble, and they better restore that, or rapidly the decay will get to the pulp chamber where the tooth nerves are, and then you'll need a root canal, a much more serious process. I'm going to make reference in some of my charts to something called the DEJ. That's the dentino-enamel junction between the outer enamel and the dentin inside. And it's literally a line that I use in my calculations, and I have to find that interface very carefully. And there's another point called the cementino-enamel junction, which is right here, which is where the enamel ends. And in that case, I need to make sure my analysis is on the proximal surface and doesn't go below the CEJ. It's a sensitive area, and it's one of the areas where you can get false positives. People who floss a lot and very hard tend to cut grooves into the enamel down here near the CEJ, and they'll look just like tooth decay in my analysis, but they're not. So that keeping track of that region is very important. This is the development process for the initial device. First off, it was a local analysis of each tooth surface, and the doctor had to start the program on the surfaces. And the first step was to find the tooth edge and the DEJ in the interproximal region, which is between the CEJ and the other thing I didn't mention. If I can go back, whoops, went back. Yeah, and the occlusal ridge, which is up here. This is the occlusal surface. We don't work on that surface. That can be done visually, and you don't really need Logicon. So we want to open up from roughly here down to there is where we're analyzing. We will look for the change in tooth density in that region due to the demineralization of the tooth from the decay. And we look for a pattern, a specific pattern that I will show you. This is really a pattern recognition program. And we calculate the probability that decay is present by comparing to a database of known caries disease, which we developed as a part of this project, and I'll show you that. And then we recommend whether the doctor should restore the surface, and we make it possible for him to save the results, either for patient education or insurance companies if it's needed, or tracking caries development over time if he doesn't restore the surface on the day he first finds the problem. I'm only developing software for finding tooth decay. The actual device for collecting the radiographs and for displaying them is completely separate. It's manufactured today by the company called Carestream Dental. This is an example of one of their sensors that you put in the patient's mouth in place of a piece of film. It's rigid, so it's a little bit uncomfortable, but it has a wire that plugs into the computer, and within a few seconds, when you take an x-ray, you get a picture that looks like this. So, and then there's an icon to launch the Logicon software. The Logicon software is a completely independent software program. Not every doctor who buys a Carestream digital sensor and software buys Logicon. If it's not a general practitioner and he's not interested in restoring teeth because of tooth decay, he doesn't need Logicon. So let's open Logicon on the example image here, and this is now a Logicon screen, and you can zoom in on the surfaces of interest. And I note in this example you can kind of see the decay on this surface already, pretty visually obvious, but it's not obvious on the contact surface. And these are the kinds of clues that a doctor uses. Any good dentist will know this is a problem and this is likely to have a problem. But the trouble is, you show this to the patient and say, well, we're going to restore both of these teeth, and the patient would say, well, I don't see anything over here. Why do we have to do that? So this is where Logicon plays a really important role. So going to the next step for the doctor, in order to launch the Logicon analysis, first he has to pick tooth type. All my algorithms are tooth type dependent. There are 4 different tooth types. I think you probably all know what they are. And then you use a custom tool that we developed to open up this V on the interproximal surface. You click with your mouse in the middle of the tooth near the pulp chamber, and you drag the mouse towards the middle of the interproximal surface, and this V opens up gradually as you move the mouse, and you position it between the CEJ and the occlusal ridge. Then you let go of the button on your mouse, and the program runs automatically, and you get this result. There are 4 diagnostic screens. You get an outline of the decay on the radiograph, which you can see is right adjacent to the other tooth where it was obvious, but boy, even with this outline, it's hard to see what it's found. You get a plot of the change in density between these red lines, and you get a probability that this is decay. Let me go on and I'll talk about each of those screens separately a little bit more. First off, this is maybe the most important result. The doctor can see the DEJ, the dentino-enamel junction. He can see that this decay has entered into the dentin. You can even begin to see some of the darker material here that's right adjacent to the other tooth where you'd expect it because the food was jammed between these 2 teeth. The trick in this software program is stated right here. It analyzes many more shades of gray than the eye can see. The Carestream sensors are 12-bit sensors, so you get 4,096 shades of gray. The human eye, first off, the average computer monitor, and believe me, dentists have average monitors. They don't want to spend big money on expensive monitors, only shows 296 shades of gray. And the human eye can only resolve 30 to 50 shades of gray. A lot of studies have been shown to show that that's true. And the things that affect how well you can do this with your eye is, number 1, the monitor. I mean, the better monitor helps. Number 2 are the background lighting conditions. So crucial, it's unreal. If a doctor was smart, he would do all his work in a dark room or with all the lights out in his operatory and close all the curtains or don't have any windows. That would give him the optimum conditions to read these radiographs, but doctors simply refuse to work that way, and patients, generally speaking, wouldn't agree to sit in a dark room and be worked on. So the ambient light conditions make a big difference. And then the third thing that makes a very big difference is how much time the doctor spends on this. If you stare at a radiograph for a while and look at all the clues, you'll probably make better decisions than if you spend only a fraction of a minute and go on. And in dentist's office, because procedures like treating teeth for tooth decay are not big money-making things, patient flow-through is really important, and the doctor is not going to spend a lot of time. That's just the truth of the matter. So all of this leads to those poor statistics that I showed you early on of doctors' success or lack thereof by just visually diagnosing tooth decay. So there was a real role for this Logicon product, believe me. Moving on here, this is the density plot, first off, the edges of these density dips are the red lines back on top of the image. And what we're looking for, the dips in the density are created by the demineralization of the tooth material from the decay. And we're looking for a pattern of density dips that are interconnected. They do not have to be in a straight line, and they generally are not. But they can't be randomly scattered around in this region that I analyzed. If they're randomly scattered around, frankly, it's not tooth decay. Something else. And I rarely ever see them randomly scattered around. The little red dot is simply a visual aid. It's in the middle of each dip, and we show the red dots when we believe there is a pattern of dips the doctor needs to be concerned with. And there certainly is in this case. And finally, this is the probability that decay is present in the enamel and the dentin. This is determined, it's really a correlation factor, maybe it's a better term than a probability, by comparing the features in this new image to a database of images that we developed at the UCLA clinic, and which I will show you more details in a minute about that. It was a major part of developing this product. And then there's a yellow line here that we call the decision threshold. When the probability in the dentin exceeds the decision threshold, we recommend the doctor consider restoring that tooth. The database is described right here. We started with 2,000 extracted teeth out of the UCLA tooth bank, and there were all types of teeth with a wide range of decay problems. They were x-rayed with film and several different commercial sensors that were available at that time, and then we cross-sectioned them and measured the depth of decay under a microscope with a tactic probe. That is really a tedious operation, let me tell you. Very tedious. It took the better part of about 18 months to do this, and we had to have 3 dentists evaluate the cross-section teeth to estimate the depth of decay, and they had to agree for us to use that data point in our database for this software. We ended up with 600 points in our database from 2,000 teeth, because that gives you an idea how difficult it is.
How many times did they disagree?
Well, they disagreed for 1,400. Okay. I mean, they did 2,000.
Right.
Unless the tooth shattered when it was cross-sectioned. And I don't remember how many shattered, but that did happen occasionally when they were cross-sectioning them, which means that's just a lost data point entirely.
But they were prone to the plus or 23% error and the 20% errors of diagnosing or false positives and false positives.
I'm sorry, I don't understand.
Oh, I'm sorry. So they fell prey to the errors, the diagnostic errors that you reported before.
No, I don't think so. They were using they were using a microscope, and they were using a probe to find where the material was intact and where it was not. So I think their process was highly accurate.
Okay.
I don't think those statistics, they weren't doing this visually. They didn't look at the radiographs. They didn't have access to the radiographs.
Right.
Yeah. They didn't start with the radiographs. They had no idea what was going to be in these slices that were cut.
So what was the source of variation?
You know, I don't know. That's a simple answer for right now. Let's talk about that a little bit later. I mean, it's not a simple process, and it's probably related to how diligent they were and what they were doing.
This process is what we call today collecting big data. Big data is everything.
Right. Anyway, then we we analyzed the density dips from all the radiographs, and then we did a fit with this database using an optimum set of parameters, which I'm not going to, they're all related to the characteristics of the density dips. You'd be surprised how many there are. It is the width, the depth, and alignment, generally speaking. And that's a bit of a trade secret at Carestream, so I'm not going to tell you which 6 parameters we ended up using. But it was very important because a number of different sets of parameters didn't work. Okay, so it wasn't a simple choice. The decision threshold is based on 15% false positive, which Professor White at UCLA told us was a reasonable figure for dentists to work at. I mean, they're not going to be perfect, okay? And it turns out to be roughly an optimum point. And it's interesting. We designed the software so the doctor could actually adjust the false positive rate. And you know, to this day, I've never met one dentist who bothered. They all believe in the 15%, and you'll probably see why in a minute, and they've never adjusted it. The only people who adjust it are academics who have other theories of things they're trying to prove, so they play with it. But
FDA didn't question them.
No, the FDA never questioned it. Yeah, we'll come to what the FDA questioned. You'd be surprised what they question and don't question. At least I was surprised when I went through the process with them. Anyway, this is the logic behind 15%. This is a receiver operation characteristic curve. These are very traditional for evaluating the relation of classified data. And I've plotted true positive versus false positive. And I told you at 15%, it's roughly an optimum point. Well, that's the knee in this curve, where you sort of have a slope of 1. If you're a very conservative dentist and you say, I only want to be wrong 5% of the time, the problem with that is that you're not right very often. You miss a lot of cases of caries. But if you're a very aggressive dentist and say, I'm willing to be wrong 30% of the time, you don't do much better than using 15%. The curve flattens out very badly. And so that's the logic in the 15%, and it stood up for years. And that affects the height of my decision threshold. If you're very conservative, the decision threshold's very high, which means you need a high correlation or probability in order to recommend restoration. If you're very aggressive, it's low, and then it's much easier to reach that decision threshold. And we're at 2. This is examples for molars. All right. Moving on, let's talk about the FDA approval of this product. Sort of interesting history. At Logicon, we studied all the procedures for applying for approval and thought sure we could apply it under a 510K. We were going to use as a predicate device, which is what you need to go 510K, the dentist himself.
Yes.
And you know what? The FDA said, no way. It was it was very interesting because in a meeting at FDA headquarters, the whole division, the radiological division that was there, 12 or 15 people. And most of the younger people in the room thought, sure, the division head was going to let us approve our product on a 510K. And the man sat there and said nothing for a good hour. And we all talked about the product and the nuances and how it works and all. And at the very end, he sort of told everybody to be quiet, and he with a very light voice announced, there's no way we can approve this as a 510K because there is no legitimate predicate device. They cannot use the dentist as a predicate device. And therefore, we had to do a pre-market approval and a clinical study. So even though the risk to patients associated with this is nowhere near what it normally is when the FDA requires a PMA study. Usually it's for life-threatening diseases and things. This certainly doesn't fall into that category. It's interesting that legally in their requirements, it's not life-threatening. What makes a PMA, it is predicate device. And
you wouldn't argue this is low risk technology, so therefore it's 510K.
Yeah, they wouldn't accept that argument at all. They wouldn't even hear of it, wouldn't even talk about it. The division head said, no way. No way. By the way, in the end, it served me very well, because that's one of the reasons no one's tried to copy this device. You might wonder why someone, I'm going to talk about the patent here, we got a patent too, but let me talk about that and I'll come back to this point. We did get a patent for this, but that was also very interesting. Namely, we submitted a patent application and it was denied. And we got a detailed letter back citing various things in the literature that the patent inspector thought had the same procedure in it as ours. And he was just wrong on every one of them, badly wrong. So we requested, we had a very smart patent attorney, and you always want a smart patent attorney. He said, don't write back. He said, we'll get in exchanging letters forever. He said, we're going to do an oral defense of this. And we went to the patent office, and I did a stand-up presentation like I am to you, to the staff of the patent office about why we thought the procedure was unique. And let me tell you, they absolutely agreed. And not only that, while I was flying home from Washington, they sent an email announcing to Logicon they were going to approve the patent, and the actual approval came through a few weeks later. They had to get different people to sign off on it in the agency. But if you really believe you got a unique process, go stand in front of them and defend it. I think that advice was really, really well given. But, so the product, no one has been able to duplicate the product for two reasons. One, we have a patent on it. But the second thing, which I've always felt is more important, is the PMA approval. For anyone who develops a competing product, they cannot use a PMA product as a predicate device. That's not allowed. If there's a product already at a PMA level, it automatically means a competing or new product that's similar has to be a PMA, period. That's just an FDA regulation. And the patent on this product, in fact, is a 20-year patent. It's going to expire soon. But I don't think Carestream's at much risk that anyone is going to try to reproduce it, because they're going to have to do a PMA. Believe me, they're expensive. I was lucky I worked for Logicon and they had a big budget, because when we did this application back in the 1990s, we spent $250,000. Today it would be $1 million or more. And it was not a complicated product. And the other interesting thing is about what the FDA worries about. You know, they ask very few questions about the technology. That's not the way they did the review. The things that they review are the results of your clinical study, whether or not you had a truly unbiased database. They worry terrible about that, because it's easy to jury rig these things with certain databases. And the labels afterwards, what claims you make. You got to be really careful with claims. If you get to be cute about it and try to tack on a lot of little things, you'll never get approval. And that's one of the reasons why at the beginning I showed you a list of limitations for this product. Those are very important. You might wonder why it doesn't work on primary teeth. And you know, it probably would, but we can't advertise that. That's an off-label use. And we didn't do an evaluation of primary teeth. Part of it being we didn't have enough primary teeth to do it correctly. So anyway, let me move on here now quickly. This is the summary of the results of the clinical study to get this approved. And it's a very simple chart. I left off all the statistical related information about errors and stuff just to keep the chart simple. But you can see how the sensitivity or true positive changed from before using the software to afterwards, 20% improvement. And it's not 100% right, but it's 90%. And then the true negative or specificity is essentially unchanged. That's really important because you can push your true positive rates up if you're willing to misdiagnose more healthy teeth. That's a trade-off you can't do. The FDA won't allow that. And the board of radiologists at the FDA, I presented this to a board of 12 very prominent radiologists in America, and they were really impressed with a 20% improvement. And I'll show you that this result is highly dependent on the patient database, and I want to emphasize before I go on to show you what happened when I changed the patient database, that this was patients that walked into the dental office on a first-come, first-served basis. They may have had no caries problems, they may have had minor caries problems, or they may have had terrible caries problem that anybody could have diagnosed without my software. We had no control over that. They wouldn't let us control that. But, and this is, whenever you get a product approved, the FDA makes you publish something called the EPI, and this is it. It's distributed with the product, and it just has the conditions in it that I told you that we worked with when we developed the product. That was why it was important to make those decisions at the beginning, so we didn't waste money on things we weren't going to get approved. And we had very good advice from Stuart White as to what would make a good product and what not to bother with. But this is an interesting chart I want to say more about. This is a second clinical study that I did 13 years later after I got this approved. That first study lasted, it was a very good marketing tool. That chart I showed you a few minutes ago, every sales rep at Carestream used for years. And then finally, I would show up at marketing meetings and they would say, Dr. Gakenheimer, can we do a newer study just for fun? We keep using this one. And I said, okay, we'll do a new study. And I worked out an arrangement with the dental school at the University of Louisville to do a study there with 12 raters, which were actually practicing dentists in their clinic. And this is the results. There were 17 images with 28 confirmed proximal dental caries. And we'll just look at a couple columns here. I'm not going to go through this whole thing, but you look at the mean value. And I didn't mention, we eliminated all the obvious cases. I only wanted it because the dental reps, actually, sometimes you do things to help the marketing guys, okay? They kept saying, we don't want to run Logicon on something everybody can see the decay. That doesn't prove anything to a dentist, and it doesn't get them to buy it. So I said, okay, we'll do a study on things you can't see. So this was literally decay that wasn't visually obvious at all and would have been missed by many dentists. And in fact, if you look at the mean value of the performance, we only found to 30% of the cases, the raters only found 30% of the cases visually, looking at the radiograph. And they used some filters in the product that didn't help them much. But when they ran the Logicon density analysis, they more than doubled the number of cases that they correctly found. And if you look at things like the standard deviation and the coefficient of variation, both are much smaller when you use Logicon than when you do anything visually. This is a really compelling argument of the value of this product. And at the same time, the true negative rate was essentially unchanged. It changed a few percent, but not really much at all to be a problem. Now, this was a fabulous result. It was better than I even expected it might be. And this was done, by the way, totally independent of me. They actually had a graduate student, like all of you, who ran this whole program. And they did he had to make arrangements for these 12 raters to come in and diagnose all these images, and this young man ran the whole show in Louisville. I was home in California, and it really is a nice result. So I just summarize a couple key points here. Clinical studies are very highly dependent on the database you use. And you need, if you have to do one for a product, start out by discussing with the FDA what they will accept and what type of database. And I will tell you right now, as much as this may surprise you, they won't be impressed just because you picked all the hardest cases. And there's an important reason for that. They want to know if you're going to screw up the easy cases by using your device, because that's a risk with the device. And that's their argument, and it's a simple argument, and they're right, okay? You could have a device that works on every hard case and then blows it with the easy cases and misleads the doctor. So that's the argument. It's no more complicated than that, and it's basically a true argument. It's a good argument. So I'm going to go on. I think the rest of the things I wrote here are obvious. In this day and age, you need patient consent for everything. If you do a clinical study that's chairside or with the patient actively involved during it, you have to have patient consent. You know, I haven't said a word about imaging filters to try to bring out the decay better. We have them. We even have a sharpening filter that we designed ourselves as a part of this product. It's another trade secret. Nobody knows what it is, but it works fabulously. But we always analyze the raw data file, and there's an important reason for that. Anyone who knows how a sharpening filter works knows that the way you make something look sharp is to kill shades of gray. I mean, they all work that way. And we want the maximum number of shades of gray in our analysis, not the minimum number. Now, mind you, when I have to find the DEJ and the tooth edge, I actually run the sharpening filter on that image to find those two lines. But then when I do the density analysis, I use the raw image. So I use the best of both worlds. This is a very interesting tool that I'm proud of that was put into the product a few years after it was on the market. The way that I've described that it works to you so far is a doctor has to open that V-tool on the interproximal surface, every surface in the image. Not only that, when he opens it and runs an analysis, he gets one result, and I showed you the one result. If you run it again and move the V into a different position slightly, you get a different result. Or you can. Depends on the decay pattern. And there are various reasons for that I'm not going to dwell on today, but the result is sensitive to where you place the V. So we would always tell dentists that they needed to run the V on a surface 2 or 3 or 4 times to be sure they found the maximum decay pattern. Well, in thinking about it, I realized, doggone it, we can do all that automatically. Why am I telling the dentist to do this? Why don't I just get my software engineers to do it? So I invented this tool, which actually automatically moves the apex of the V around, and I chose 9 points in this yellow box. Now, I had a very interesting criteria for defining the box and also for using only 9 points. I won't go into that today, but I spent a lot of time thinking about that. It wasn't any accident that I arrived to this. And then the arms of the V tool are held fixed near the CEJ and the occlusal ridge. So you keep those fixed. So you're changing sort of the angulation around this decay pattern, and it's the geometry of the decay pattern. If the decay pattern were a straight line, none of this would matter. The problem is when it's not. Then it starts to matter. Anyway, I automatically do 9 calculations and pick out the best result, which is in general the deepest extent of the decay. Great, worked perfect. Then I decided, because I had a few dentists who were so picky, that I would also make it possible for them to click in the middle of the yellow box, open up the V, and drag the apex around to any point they want. And then the calculation updates in real time. I mean, the computers are fast enough that it updates in real time. People love that. So these guys who want to just find every little density dip everywhere were dragging the apex around inside the yellow box. Wouldn't let them out of the box, okay? But this was a very interesting result, and this came from worrying about workflow and talking to doctors and realizing that the original way we designed this was too much work. And the net result was some doctors used it religiously. They usually had their hygienists do the work for them. But other doctors didn't use my product because they said it just didn't work in their office. It took them too long to do it. It's very important development. We also have the capability to draw the tooth edge in the DEJ, and there are special cases like when teeth are overlapped. You can actually analyze the region that's outside of the overlap by drawing the position of the lines. If I tried to use the V, normally it would include the overlap, which I didn't want. And also, if there was a bright amalgam near the surface I was trying to analyze, it frequently distorted the V-tool calculations. So again, we drew the tooth edge in the DEJ. Tech support. So I've told you about what I call the first version of the product with the addition of the ROI tool. There's another version that I'm going to tell you about. And it came about because one of the things I did as a developer, and I manufactured this software for the distributors. So it's all it involved is duplicating a CD. So not too hard. I did all the tech support, and I insisted on doing it. I had a lady who worked full-time for me in my home office, and her job was to talk to dentists, and any serious problems, she would get me involved. She was very highly skilled, and she didn't need me most of the time. But once in a while, doctors would call up either very upset or with a very unique problem or both, and I would actually take the tech call myself. And one thing it became very clear to me, if I wanted to raise this product to a really big-time way, I had to automate all of this because it was still taking up too much time. So that's what happens. And I've listed all the tech support things I was involved in. I'm not going to go into them in any detail. You can read them. I did a lot of webinars. I did webinars like once a month, and sometimes I'd have 50 people on the webinar in different dental offices all over America. They had bought the product and they wanted training from me. And I trained the Carestream trainers. Carestream had a great team of trainers, and I was very proud of their trainers. They really showed Logicon well. But some offices didn't buy the training because it was expensive. So that's when I did webinars. Anyway, the feedback was it's still a workflow problem. It's too slow, and we need to fully automate the scanning process. So I invented a feature called Pre-Scan, and it was released in 2016. This is the engineering development process for that, and you see the word automatic on almost everything. First, we had to segment all the interproximal surfaces automatically. See, in the original product, the dentist designated where that surface is. And I had to identify the tooth types. The dentist was selecting the tooth type, but my algorithms are tooth type dependent. So we needed to recognize all the teeth. Then we needed to open the V tool automatically. We didn't have the luxury of the doctor doing it. Then we did run the ROI tool and get 9 results and pick the best one for each surface. And then we had to review. We had to take that file on all the surfaces in the radiograph and review the results and discard some that we knew were not really tooth decay. So this is really making this an AI product and smart software. And that criteria, let me tell you, I worked on for a year. And I had so many conference calls with my engineers in Taiwan, you can't believe them, because they were having trouble with it. And I would see that it was obvious things to throw out, but they couldn't think through how to do that automatically in the software. So I had to really help them, and we succeeded. Believe me, we've succeeded. But this took 4 years, and I had 4 engineers in Taipei working on this. This was a bigger job than the original product, by a substantially bigger job. The big part of the original job was developing that database at UCLA to do the probabilities. Quite frankly, the image analysis was very straightforward. It was actually easy. Okay, it really was. But this was not easy. And to get the error rate down to a point that it would be acceptable was an awful lot of work, to tell you the truth.
Did you anticipate that difficulty?
What?
Did you anticipate that difficulty?
Yes. Oh yeah. Oh yeah, I did. Yeah, I did, to tell you the truth. Anyway, so now, what do? You click on one icon, and it takes 10 seconds to 15 seconds to do all the surfaces in this image. And here's the result. And you get a simple code that goes with this. Anything with a red outline is a surface the doctor should consider restoring. Anything with yellow are surfaces he should watch carefully, but probably don't need restoring today. And an X...
I'm in trouble. I see so many red.
Yeah, this patient needs help. X's mean it didn't scan. For example, I think all the cases in this image that didn't scan is because there's restorations in the way. And we just needed to mark those. If there's no mark on a surface of any sort, it actually means it's a clean tooth. And it did do an analysis and save the result for clean teeth. And I'll show you how you find those in a minute. But the result for every surface is saved here. Okay? And so I'll show you some of the details of the results, but I'm going to diverge quickly to the FDA approval process for this, because this was an interesting experience for me with the FDA. And by the way, I had great relations with the FDA. And I enjoyed working with them, and I think they added a lot to my product. You know, some people will disagree, and there were people, you saw Eastman Kodak that was on my list there as distributors. By the way, Eastman Kodak bought Trophy, the French company, and then spun out Carestream. I think I said that. In any event, some people would take on the FDA as opposed to listen and work with them. Big mistake to take them on. Big mistake. They get brownie points for approving products. Their job is not to not approve your product. Their job is to be sure it's safe. Okay? And they do their job very well. And in any event, what I had been doing for all the changes in my product, like that ROI tool, was simply reporting it in my annual report. And I'll show you that page of that annual report in a minute. And the only situation that a supplement is required is if the safety and the effectiveness of the device is affected. I didn't really think it was, so we had gone ahead and started to distribute the product. And then when they heard about it from my annual report, because that's the first time they heard about it, they disagreed. and the person who reviewed the report said, I think safety and effectiveness might have been affected here. You need to do a supplement. So we stopped distributing the product, and I did a supplement. And I had done an in-house clinical study that supported the version that I released. You always have to do that, and you always have to have it in your filing cabinet. I did a clinical study for that little ROI tool and had it in my filing cabinet. Well, I submitted quickly that study. They looked at it and said, no, we won't accept that, and I'll show you why in a minute. And then we had a conference call, and they proposed a completely different study, which strangely enough doesn't really involve a clinical study at all. It involves comparing the new automatic device to the approved original device. That was actually much simpler than the clinical study I did and put in my filing cabinet. Much simpler. But it was kind of interesting that sometimes, this is why it's wise to talk to them, okay? Because they actually simplified the whole thing. Anyway, and then I had to recall all the early devices that I had released before I did this supplement. But because we copy protected our software, every user has to get a site key. It was easy to go back and ask them to change the software. Just quickly, this is my table that I annually submitted, and it was a rolling table. Grayed out things were submitted the year earlier, and they approved them, and then the new things were always here in white. They always like this. They like to see the whole history as they're reviewing a product. And then here's pre-scan, which they did not approve and asked for a supplement. This is a summary of the clinical study I did to support the development of pre-scan that was in my filing cabinet. And this is a little bit complicated, but if you look at the column for true positives, and if you look at these numbers there's a pair of numbers here. The first number is running the automatic software without the doctor interceding at all. Just he accepts the result. The second number is when he corrects some of the calculations, and we had very simple criteria for him to correct them, like it didn't run at all. That's a no-brainer. If you got an X on it, but it could have been run, he can go back and do that one. Or if the carries pattern is incomplete, he can go back and correct the carries pattern. In any event, you see what the performance was for true positive. I got it up to 96% with the doctor correcting it. And the true positive, true negative rate was 94%. These were great numbers. So what's wrong with the FDA? They didn't like the way I chose the patient cases that I did. And I chose only difficult ones. This goes back to the same thing that I mentioned before. They said, great, you got all the hard cases, so what did it do to the easy cases? Okay, so I hadn't included those. So when we went on and we did this confusion matrix, which can also just be called a comparison matrix, but the FDA likes the word confusion. Don't ask me why. We randomly selected patient images and without any consideration of the degree of caries on them. And we analyzed them all with the original approved version, which is called 4.1. Then we ran the new automatic version. And where things agree are along the diagonal. If the numbers off diagonal don't agree. And if you calculate the overall accuracy, you get 78%. And this is if the doctor does nothing. If you let the doctor correct, if you go back and look at this, these cases didn't scan. Basically what the doctor did when we let him work on this was he did all the ones that didn't scan. And then you see that the accuracy actually went up. And they loved this result. They said, just fine. You can, now you can distribute it. Again, taking care with what your patient database is when you do a clinical study. It's so important, it's unreal. Because in a big clinical study, a lot of time, years, can be lost if you have used something they won't accept later. It's we had the capability just to toggle the doctor. The whole idea here is the doctor has to review these pre-scan calculations, because if he doesn't, suddenly my device became a concurrent device instead of an assistant. So what I'm showing you with the rest of this is, now there are two screens. There's a pre-scan screen, and then there's another screen where the doctor saves the results he wants to use in his treatment plan. And he can also toggle things off. So I just took that image where I ran pre-scan and toggled all the lines off, so now the doctor can visually look around. But here's the results right here. Secondly, if he wants to see the details for any surface, he just clicks on the surface and the details appear. And he can, and then I jump by this box here. He can decide whether or not to keep it in the doctor's diagnostic screen. So save it, yes or no. Then this is one where the caries pattern was incomplete, and he can choose to try to adjust this pattern. He gets a warning message about it. See, what's happened here is decay almost through the enamel. Then there's some area in here where there are no density dips, and then there are more density dips in the dentin. Well, if these density dips in the dentin were way over here or over there, I'd say they have nothing to do with this caries pattern. But because they're lined right up with it, you know very well they do. And what's happened is the density dips were probably very small in this region, and the software couldn't find them, maybe because of the noise in the radiograph. And so he can try to adjust for it. He can open up the V tool. Remember I told you can drag this thing around, and you can actually look for the missing dips. And I think in this picture right here, I found one more dip, and the probability is a little bit higher. And he can save that result. So this is the list of pre-scan results. These are the ones he's chosen to save. There's a few more. And this is the doctor's screen. This might be what he shows the patient, and this might be what he uses to justify his treatment plan. And see, it's a much smaller number of surfaces. Now this is not a concurrent diagnosis. This is a doctor's diagnosis assisted with the software. And the last few things I've got here, we have a quick start guide and a user guide embedded in the software. We have help files to explain how to open that V tool, for example, and how not to open it. There's only one correct way here, and all these other ways are incorrect. And how to use the pencil tool. And I even developed a training wizard, which is step-by-step questions. You know, you start the software and a question box comes up, and in order to go on, you have to pick the right choice. And it actually walks you through a diagnosis. Mind you, most people who bought my product, I never got to meet face-to-face, 95% of them. And training them with this has always been a little bit of a challenge. This is a very slow process, but very useful. You'd be surprised how many doctors who bought the product didn't even read the EPI. Always used to make me mad. Read what it's supposed to do for you before you call me up and tell me it doesn't work. They were trying to use it on things that we never had it cleared for, and they weren't supposed to use. And my closing chart. I think this is a very good early example of how software and AI can really better healthcare. Now, a lot of people are learning that. There's a lot of other projects going on now here, and many universities around the country. Johns Hopkins, where I got my bachelor's degree, is doing a lot of work. Their School of Engineering at Johns Hopkins, I think close to half of it works with the medical school now. It's really, that is a tremendous application, association there. And, you know, with the improvement in computer power and memory and the growth of patient databases, there's just a huge opportunity now to combine engineering and software and to help improve medical diagnostic devices. You know, the few things that I've identified as really lagging and is the development of streamlining user interfaces. A lot of things, they're overly complicated or overly vague. In other words, you don't really know how to use it unless someone shows you. Quick start guides for many products are terrible. You know, the quick start guide that came with my Dell computer that I'm using up here is worthless. It's worthless. It's the stupidest thing I've ever seen. And I had to learn how to set up my Dell computer by calling Dell and having somebody in India walk me through the whole computer. I mean, this is terrible. And building in training tools and a friendly, detailed user guide that you can quickly research, and warning messages when you really do make errors. You know, people don't like warning messages. It's interesting, because if it's wrong, they consider it a real nuisance. And you'd be surprised that doctors will ask me to suppress the warning messages. The trouble is, I don't agree. First off, I admit that occasionally they come up when they don't apply, but that's actually not very often. The other side of it is, it saves a lot of mistakes. And it's another value of having a software product. And with that, I'm done. Thank you all.
Thank you, Gakenheimer. This is a very detailed speech about the success of a story. How many of you read your cell phone manual? None. See?
Right.
All right. We have a little bit of time. Maybe two quick questions. Anyone has any question for Dr. Gakenheimer? Go ahead.
I will. So it was interesting. So you hired a really good patent attorney for the IP, but you did it yourself as far as the regulatory affairs went?
Right.
So the question for sort of the general biomedical entrepreneur, do you still recommend that? Because what was interesting is you said you lost a year of time to market pre-scan. And that could have been the end of a company.
That's a good question. First off, very good question. Let me emphasize that you need the patent attorney once. And you either get the patent or you don't, and that's the end of it. In terms of the regulatory expert, you need them not only to follow the original application, but to help you monitor the product through its life cycle. Now, the initial decision that we thought a 510K applied, we did confer with some people that we thought were very knowledgeable in the area at UCLA. And I also told you that almost the whole staff of the radiological division that was in the room thought it applied. It was only their director, and I think he pulled it out of the hat. I mean, I think they had not been confronted with something this unique. Okay? Now, as time went on, after it was approved, we did lose 2 years. But once we got it approved, I did hire experts to help me write all the documentation I needed in my office. The, what are they called? The there's a set of rules that you have to write for manufacturing and controlling your product, and the SOPs, standard operating procedures. I had 30-some operating procedures for this product. I had helped to write those because they greatly expedited getting that written.
It's a GMP, good manufacturing.
Yeah, and then we did our own in-house inspections. I did 4 a year. And to be sure, when a real inspector like the FDA came in, that most things were in order. But you know, I had a small company. For me to hire all those people and and pay for that, it was expensive. And even at Northrop, when the original or Logicon, when the original application was filed, we consulted with some people. But you know, I had a lot of engineers working for me. They wanted to use them. But you're right. If you really, you know, if you're in an institution or somewhere where they have the resources to hire people, it's probably best to hire them. The other problem that I found is it's a job by itself to educate them on what you're doing. Okay, the patent attorney was actually a good example of that. He was very difficult to explain the product to, and it took a long time. But he was very smart to know to do an oral defense. A lot of people don't know you're entitled to an oral defense. I have discovered that. And it's actually in the patent law. You're always entitled to an oral defense.
Well, without IP, you may have a problem to raise money, right? So investors will walk away. So to me, it's very important to have some IP. Today, at least for today.
You had a second question?
Do we have a second question? No?
I can make one comment.
Okay.
Sort of having come out of the radiology.
Okay, the last
word. I got the last
Okay.
word. So I came out of the radiology world and actually did image quality assurance. And I want to just second what you said, is that all the training and all the support stuff is enormously important. And that's called the whole product. So as you're sort of going into doing product development, if you're a technical entrepreneur, do not undersell that. It's, as you said, it's
Right.
very important, very difficult.
Right.
A lot of work.
Okay.
Thank you.
Let's thank Dr. Gakenheimer.
Thank you all.
If you come